Hello everyone, I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Rapid Deployment of Custom Multiplexed Assays for Clinical and Translational Research Laboratories, and our sponsor is Agena Bioscience. Our panelists today are Connie Lisle, Senior Lab Manager at LifeLabs Genetics, Dr. William Shek, Senior Scientific Director for Research Animal Diagnostic Services at Charles River Laboratories, and Priscilla Hunt, Global Product Support Manager at Agena. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you do not see a Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentations have concluded. Ms. Lisle, please go ahead.
Good afternoon, everybody. Oh, I don't see the presentation.
Connie, are you having a problem accessing your presentation?
Yeah. Yes, I am actually. I can see my picture, but I can't see the actual slides.
Say next slide and we'll hit it for you.
There we go. Okay, wonderful.
Okay.
Thank you so much. Good afternoon, everybody. Today I'll just be talking about customizing assays for the mass array. Next slide, please. I am an employee of LifeLabs since 2015. I have been supported and asked by Agena Bioscience to make the presentation to you today. I'd like to acknowledge my technical team at LifeLabs Genetics and special acknowledgement to the Agena Bioscience team that supports us throughout our assay builds, Julie, Robin, and Hema. Next slide, please. LifeLabs is Canada's largest community service laboratory provider. We have over 5,000 employees. We have more than 375 specimen collection centers spread out through Ontario, BC, and Saskatchewan. We currently perform more than 100 million lab tests per year, which services 11 million patients annually. LifeLabs Genetics was created as a new molecular genetics service in 2014. We received our clinical laboratory license, IQMH, and ISO 15189 certification in 2015. We were CAP accredited in 2015, and our CLIA application is pending. Next slide. Our clinical test portfolios include reproductive health. We do NIPT and expanded carrier screening. We test for hereditary and familial conditions, and we provide pharmacogenetic and wellness testing. And it is for this area of our portfolio that we've adopted the Agena Mass Array platform. And all of our assays are SNP-based testing, and we provide these for clinical partners. Next slide. Our decision point for choosing the Agena Mass Array, it has a very low reagent cost per analyte compared to other platforms, a low labor cost, and a very easy workflow to incorporate into your lab. The processing is quite rapid. We can combine all of the assays in a common single protocol. It's easily scalable, which gives us great capacity for doing a lot in a service lab, and it's a highly reproducible platform. It covers a range of assay complexity. It's not limited just to single nucleotide polymorphisms. We can actually examine multiple different variant types like CNVs and indels. It's effective for regions with high sequence homology, like HLA. We can use allele-specific strategies for a selection of targeted risk alleles, and we can quantitate CNVs on this platform, for example, your CYP2D6. It has great flexibility. It offers flexibility in adding or changing assays. and when that's done, you don't always require a large-scale revalidation because each well can be validated independently. We can combine up to 35 analytes in a single well, and we can combine different assays in one run because all of the protocols are the same. It's a useful platform for different test applications. Agena has pre-designed kits that are ready to use. They have particular expertise in PGX, which was of interest to us. We have the ability to custom design panels, and they offer assay development options like an online assay design tool and their Assays by Agena custom assay service. Next slide, please. Assays by Agena is a service that's offered by Agena to assist in customizing your assay. And we chose to use them because they are experts in complex assay design, in the mass array technology, and in the chemistries. They take care of the design, optimization, verification of the primers. In optimizing allele separation, they can handle your sequence and design challenges. For anybody who's designed multiplexed assays, we know that there are sometimes it is quite challenging. They have related tests with similar targets to draw from, and so we can make use of that. They're a very attractive platform for a clinical service laboratory because they offer a very convenient service. They maintain your confidentiality when you're sharing the panels with them. They address the time and resource constraints that a clinical laboratory always seems to have, and they address the lack of in-house expertise that you may have in building something on the platform. They improve the time to launch for a new assay. 4 to 6 weeks is what it takes to deliver a working assay. Cost effectiveness in using this service, they provide an upfront price per assay well. They ensure that maximal multiplexing is reached, and they can provide results in the less time it would take to design it in-house, and that results in less optimization and troubleshooting time that's required at the lab. The ownership of the assay goes back to the lab, and ongoing support is provided by Agena for that product, and they can support any future revisions or updates that need to be made to that panel. They deliver high-quality products. Their products are well-defined, consistent, and highly reproducible. They operate under high-quality standards, practice reagent control and lot tracking. They do primer verification and QC. These are all important for a service laboratory. And on an ongoing basis, you can purchase pre-mixed primer mixes that are QC'd. Again, that's very attractive for a clinical service. Next slide. Our experience in working with Assays by Agena. We have currently customized 2 PGX and 1 wellness test with them. Our tests cover a variety of different variant types. We have a 2-well assay that incorporates 45 variants, a 4-well assay that covers 65 variants, and a 6-well assay that covers 164 variants. So you can see we've got quite a widespread menu of products. Within those different products that we have, there's variable complexity in our assays. They all are a different combination of SNPs, deletions, CNVs, and HLA. We use strategic designs like allele-specific amplification to target very specific risk alleles that we were after for HLA specifically. And they were able to use historical designs that they have incorporated into some of their PGX assays, which cut down some of the time that it took for us to design new ones. We had a true partnership approach when working with them. They projected timelines and provided milestones that were well-defined. We had regular touch points between the teams. We had phone calls, emails. The field application specialist made numerous visits to us. And we maintain very open communication and sharing of the data all the way throughout the process. And finally, they delivered what they promised. We're very happy with the products that they helped us design and with the service that they provided. There was minimal and targeted optimization once they delivered that first product to us. We did have, as always, you will have some optimization that has to be done. However, it was minimal. All of the assays did work under the same workflow, and so we've been able to automate some of them. And on an ongoing basis, we're able to batch our different tests in a single run, which makes for a very cost-effective and efficient process. These assays are definitely sensitive and specific, and we continue to order, for some of the tests, we order the pre-mixed primers that they provide. However, for some of them, they've been able to provide us details so that we can pre-mix our own if we found that that was more cost-effective. Next slide, please. This is just depicting the process flow for working with Assays by Agena. You'd provide a marker list to them and indicating all of the information that you possibly can, the gene, the RSID, any reference you have or transcript information, and all of the possible genotypes that you are looking to be able to target. They would design PCR and extension primers using their assay designer suite and online tools. They use NCBI database to ensure that they're using proper sequences and designing the best primers for your application. They do empirical testing and optimizations in-house with characterized samples. And then once they have an assay that's working, they would transfer the primer mixes as well as the assay design file to your lab for trial. And you would evaluate those assays using your clinical samples, any positive controls that you have. You want to ensure that that all the alleles can be distinguished and that you have acceptable peak heights. And so there's a loop that goes back. Once you've evaluated in your lab, you would provide feedback back to Assays by Agena with any optimization that needs to happen further. And then that process would happen again. They would do their optimization and again flow it back down. And that can happen a few times, that loop, until you have a product that you feel you can go on with your test validation. And the process from when you provide the marker list to getting the first working assay in your lab, for us, when we did it, it was 6 to 8 weeks. However, they have brought that down to 4 to 6 weeks now, which is really just a testament to how committed they are to providing the optimal service. And anywhere from 3 to 6 weeks is what, in our experience, it's taken us from that feedback loop to get to an assay that we can actually continue on with true technical validation in our lab. Next slide, please. This is just an example of optimization that needed to get done. So on the left would be the primer mix that we received from them as a first pass. Now, as I said, they build their assays using, they test their assay using controls. And once you bring it to your lab and you use your clinical samples, there can be challenges with your sample type. And so that would be an example of what a particular assay looked like when we got it. We fed back that information and asked for tighter genotype scatters. And so on the right is the re-optimized primer mix. Next slide, please. To give you a taste of our challenge, our biggest challenge so far with them, and I believe they probably would agree that this was up there in their challenges. it's a test that we call TreatGX Plus. It's a pharmacogenetic test that was originally developed by a company called Genexus. LifeLabs partnered with them in 2017 to bring this test into Canada. To date, it was the most complex panel we had seen. We needed to target more than 60 SNPs. We had 2 CNVs and 4 HLA risk alleles that we were looking to build in a single assay. We required this test to be more cost-effective than what it was at Genexus so that we could succeed in this highly competitive and price-sensitive market. We were under operational constraints at the time. We had a validation of this nature had a very large time commitment implication for designing and validating it. We didn't have any experience with validating something, a test of this complexity at the time. We were introducing a new sample type, and we had contractual agreement for a turnaround time of a launch. So we were under time constraints. We required flexibility in the design of this assay so that we could manage the ever-evolving state of PGX testing. Medical indications and clinical findings are continuously advancing, so we wanted an assay that we could add to and not necessarily have to change and revalidate every single time. And in fact, we were faced with that challenge partway through the validation of version 1 of our assay. ABA actually had to redesign the entire assay to accommodate some additional SNPs and CNVs and HLAs and the elimination of others. So it was a real challenge, but they took it on for us. Next slide. So the result, the original Genexus test comprised of multiple different methodologies. It was many of the SNPs were on the TACMan open array. They had multiple qPCR assays that they were doing as well, and Sanger sequencing and long-range PCR. So you required all of those methodologies to provide all of the information they were looking at. LifeLabs was able to simplify the test along with ABA to create a clinical assay that is more economical, it was sensitive, it was much more scalable and automatable. And we were able to do that by combining everything onto the mass array and keeping conformational Sanger sequencing for the HLAs. Next slide, please. Assay Design Suite is one of the other options that you have for customizing an assay on the mass array platform. Really, Agena offers online design tools for building the assays in-house. The design tools are easy to use. They can custom design sequences when you define the targets that you're looking for. They will help to validate the PCR and extension primers and provide any oligo information that you'll need for ordering and testing your oligos, as well as the design file. We actually do use the Assay Design Suite for designing small panels where it's manageable. They're under 20 SNPs, much more manageable in our hands. We've also designed alternate primers for the current assays we have, so when we have had to change out a primer here and there based on a slightly different target we're looking for, we're able to do that. And we actually use it for designing sequencing primers for our familial testing as well. What we've learned is that a multiplex that works on paper doesn't always work that well at the bench. And so although the tools are fantastic and easy to use and they offer a lot of information, they don't offer the design expertise in the software. And so it does take more time to ensure that the sequence design and assay optimizations are actually working than it does when you're using their Assays by Agena service. Next slide, please. So our lessons learned. in creating these assays, customizing our assays on the mass array, I would say overall, the time spent optimizing your reaction conditions is well worth it. You have to invest the time up front. Running with suboptimal primer designs and ineffective multiplexing increases the amount of repeat testing. And so you really need to invest the time in good primer design. Know your target alleles and any inhibiting or interfering sequences that will affect the effectiveness of your assay, and make use of Agena's assay design solutions. It's important to work with proper controls and clinical specimens to ensure that you're priming and selecting the right target, and that enough product is being generated at the beginning to enable you to get good peaks in the end. Ensure that you want to ensure that your extension reactions are proceeding to completion, and that all of your alleles are discrete and well separated. These products should give you reliable and very reproducible results. Duplicate testing should not be required. And specimen preparation is key in really getting the best quality data. We also learned along the way to keep the mass array properly maintained. Next slide, please. For future development of customized assays on the mass array, we feel that we have gained familiarity with the mass array system, the chemistry. You know, we have some experience now with assay development and the different types of variants that can go into a customized assay. Agena provides very effective training and always very supportive. We've had great success with their ABA service as well as their assay design suite to design smaller panels. We definitely know that it takes more time to do the designing in-house, but we've gained important knowledge by doing that. Sometimes it is more cost-effective, and so I think you just need to weigh out when you're designing a project, just weigh out the differences between the services that they offer. And cost and time are always a consideration for us and for any clinical service labs, so we will definitely continue to use ABA for our complex panels, as we feel they're the most efficient solution to designing a very reliable product and for getting it to market, to the testing lab very quickly. Next page, please. Thank you for your attention, and I'll turn it back over to Christy.
Thank you, Ms. Lisle. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. And next up is Dr. Shek. Please go ahead.
Thank you, Christy. Okay, so today I'm going to talk about genetic monitoring of rodent models using the Agena Iplex PCR mass array system to look at SNPs. First, I'll give you a little background about Charles River Laboratories. It was founded by a veterinarian named Henry Foster in 1947. He was a real pioneer in the field of laboratory animal medicine, and his great idea was how to produce pathogen-free or SPF animals on a large scale by a process that goes by the acronym of COBS, which stands for Cesarean Originated Barrier Sustained. And as I'm sure many of you know, the reason to use SPF animals in research is that they're free of pathogens that have been shown to interfere with research or compromise the accuracy of your research. In the mid-1990s, under the leadership of Henry Foster's son, Jim Foster, Charles River has expanded principally into the area of contract research, also in areas of clinical microbiology, including doing the LAL test and the production of SPF embryonated eggs. Part of Henry Foster's vision, in addition to having rigorous production methods, was that the realization that no process is perfect, and therefore your process needs to include inspection and laboratory testing to verify that animals are meeting the specifications that you have for them, and most importantly, that they're free of pathogens. So because of this realization, he established our laboratory, which is generally referred to as Laboratory Services, in the 1960s. And when I started at Charles River in the early 1980s, we began offering these services to external institutions. And the services that we offer today include diagnostics or health monitoring to look for the contaminations with pathogens by traditional methodologies, including pathology and parasitology, but more often today by molecular genetics methods, primarily the PCR. And in addition to looking for pathogens in animals, we test biologics. And recently, with the increasing interest in the effect of the microbiome on the response of animals to experimental conditions, we've started offering next-generation sequencing. In addition to doing diagnostic testing, we do genetic monitoring, where we look for specific mutations. As you know, there's been a dramatic increase in the use of mutant models, including those that are genetically engineered, so we monitor them to show that they have the expected mutations and also look at gene expression. But what I've been involved with more is the routine quality control of inbred strains and outbred stocks. In the case of inbred strains, we're looking for unintentional outcrosses or genetic contamination, and we also look for genetic drift due to mutations that causes genetic divergence among sublines of inbred strains, such as the B6J from Jackson and the B6N from the NIH. We also look at outbred stocks to show that they're not losing their heterozygosity, that is becoming inbred, and we compare colonies. And I'll show you some examples of these data. So I just want to talk about, very briefly, some history of genetic monitoring. The most important part of genetic monitoring is to look at qualitative genetic markers. These markers, of course, have to be polymorphic. They have to distinguish among strains and individuals within an outbred stock. Ideally, they should be co-dominant, so you can identify animals that are heterozygous at a particular marker, distributed across the genome. So when you do this monitoring, you look at a large part of the genome, and therefore are more likely to detect a genetic contamination. And also that they distinguish among strains and stocks that are likely sources of genetic contamination. So in our case, we'd want to choose markers that would distinguish among the different strains that we might be raising in the same facility or even in the same room. When I first started doing this work in the early 1980s, we looked at phenotypic, biochemical, and immunological markers. And there's a diagram here on the right showing the way by electrophoresis, according to the migration rate of these markers, that we could distinguish between allozymes of the peptidase 3 enzymes. More recently, with advances in molecular genetic technology, and other laboratories have begun looking at gene sequence polymorphisms, starting with variations in the number of tandem repeats in satellite DNA, mini-satellites, which are detected by southern blotting, and microsatellite variations, which are detected by PCR. And we in other labs have even more recently switched to relying on single nucleotide polymorphisms for our genetic monitoring programs because the SNPs are stable, they're numerous, therefore found throughout the genome. One shortcoming is that they are dimorphic. They only come in two forms, so it makes it less likely that a particular marker will be unique to a given strain. So how do we use these SNPs in our genetic monitoring program? Well, what we do when we're testing inbred and F1 hybrid strains is compare the allelic profiles we get, and I'm showing examples of those on the right side of the slide, to our reference profiles. In the case of inbred and F1 hybrid animals, we expect the profiles that we get when we test samples of a given strain to correspond to the reference profile for that strain. And in this example here, we have a 32 SNP panel, and later I'm going to talk about our transfer of our 32 SNP panel to the Agena mass array system. And this is the panel that we use for routine monitoring of our inbred strains to look for genetic contamination. It's unlikely that deviation from these profiles would be associated with genetic drift due to mutation. But we do have larger panels, which we're also developing on the Agena platform, that we, by which we can detect the genetic drift that might distinguish among substrains or sublines of an inbred strain. So what do we do with these test results when we're done? Well, we'll test a certain number of animals per area, and in our case, we do this testing quarterly. And for inbred and F1 hybrid strains, and as I mentioned before, we compare those profiles to the reference profiles. But before we do that, we look at the suitability of the sample for the testing, and we do that by calculating a call rate. And we expect the call rate to be at least 60%. You can see in this table that the call rates for all samples exceeded 99%, indicating that the samples that we tested were suitable for suitable for SNP testing. Why? Typically when the call rate is low, it's because the quality of the samples is low and they're not amplified, and therefore we can't read the results. Then we calculate the percent match or correspondence between the expected allelic profile and that of the samples that we've tested, and you can see in this case, as we generally expect, the percent match exceeds our, for inbred strains, exceeds 98%. And exceeds that too for F1 hybrids, although it doesn't always, because if we're testing male rodents, they'll be hemizygous at X-linked SNP loci that would be heterozygous in females. Okay, I'm just showing you a quick example of some of the data that we generate when looking at outbred stocks, because remember, outbred stocks are characterized by heterozygosity and they therefore don't have an established allelic profile. Instead, what we're looking for is a stable level of heterozygosity. If you look at the table on the left, you see HO and HE, which stand for observed and expected heterozygosity. The expected heterozygosity is a calculated value, and using those, the expected and observed heterozygosity, we calculate the fixation index, and what we want to see is the value for the fixation index is near zero, indicating that we're not inbreeding these animals. So our goal is to maintain this level of heterozygosity. What we also do with the information that we get from SNP testing is to compare our areas to look for differentiation among colonies, and we do this by calculating a value called F sub ST, the fixation index for subpopulations versus a total that measures a genetic distance between subpopulations, and a value of less than 0.05, and you can see the values here are less than 0.05, indicates that the differentiation among these colonies is minimal. So that's the goal of our genetic quality control program, is to maintain a constant level of heterozygosity and minimize genetic differentiation among our colonies. Okay, so now finally I'm going to talk about our experience with developing SNP genetic monitoring with the Agena Iplex PCR mass array system. The steps of the assay that we're performing are shown in the figure on the left of the slide. The first step is to do a multiplex PCR. So the data that I will show you in subsequent slides is for 32 SNP multiplex. So for the 32 SNP panel, we're doing all the assays in a single well. And in this first PCR reaction, we amplify the sequences that contain the SNPs that we're monitoring. Then shrimp alkaline phosphatase is added to dephosphorylate unincorporated DMPs, and there's an extension primer with terminator nucleotides. So the extension primer hybridizes to the amplicons that you generated in the first reaction, and then the single nucleotide base is added. And finally, the extension products are analyzed by MALDI-TOF mass spectrometry, where the time of flight is related to the molecular weight of the extension products, and therefore SNPs are distinguished from one another according to molecular weight. So why did we switch to why are we in the process, I should say, of switching to the SNP Iplex? And first of all, we had issues with the assay system that we were using, including lot-to-lot variation. And another issue was that the software couldn't be relied upon to call the assay results accurately. So I have been spending a lot of time, and others, making laborious manual call adjustments. We felt and are demonstrating that this would not, these manual call adjustments would not be necessary with SNPs that were distinguished according to their molecular weight. So we wanted a system that would be more accurate and reproducible and be automated, particularly when it came to calling results. This is a diagram that I took from the Agena literature, and they have a lot of great literature, which shows the steps of the assay, and that it takes around 7 hours to complete a run, going from the initial PCR amplification to the SAP and primer extension steps, and then using a nano dispenser to add the reactions to the spectra chips that are then placed in the MALDI-TOF mass spectrometer. The results are then transferred to the typer software, which makes the calls. The next step then is to export, in our case, is to export the data to our analysis worksheet, and we were helped with that by Agena's IT support. They generated a report that worked very well with our workbook. Okay, so what did we do for development and qualification of our mouse 32 SNP panel that I'm going to talk about now? Well, assay development, as discussed by Connie, was done by Agena, and that was very, very helpful. We provided, and again, Connie mentioned this, the same thing in her case, the NCBI reference IDs for the 32 SNP panel, and key to the process is the preparation of standard inbred and F1 hybrid specimens to qualify the assays, and we also provided reference genotypes. I mentioned results reporting is by typer software. It's really the results calls are made by typer software, and the IT department at Agena developed a CSV file that we could export after the typer software called the results. Analysis, I mentioned before, is done in a custom Excel workbook, principally calculating the call rate to determine sample suitability and the percent match to the appropriate reference allelic profile to determine whether genetic contamination had occurred or not. The qualification had two basic steps. First, we did replicate testing of the standards for SOP development, and again, there's a lot of literature that helps with that that's provided by Agena. And in addition, we had very good technical support and training from Agena as well that has made our progress possible. We're also doing this testing to demonstrate the accuracy and reproducibility of the tests without having concerns about the quality of the sample. So we prepared these standard samples. We knew they were suitable for testing. We knew that the animals were not genetically contaminated. And that allowed us to evaluate accuracy and reproducibility following the basic experimental design that's listed here, where we tested 5 inbred and 3 F1 hybrid strains. And testing F1 hybrids is particularly important because one of the issues can be the ability of the Agena typer software to distinguish between homo and heterozygotes. And we did have one instance in this panel where the distinction was not very good, and the assay was re-engineered by Agena and now is working perfectly. We tested 3 samples per strain, and each sample in triplicate, and we wanted to have at least 3 successful runs. The next step that we have completed is parallel testing of field samples, that is, samples that would be submitted to our lab, and that's a way of monitoring the robustness of the system, its tolerance for sample and tech variability. And finally, its suitability to be a replacement for the testing system that we have been using. So here you see an example of an assay, and it was just one of the 32 assays that was not doing a good job of distinguishing between homo and heterozygotes. And so if you look at the original arrow, you can see that visually you can distinguish between the homo and heterozygotes, but the software wasn't able to make that call properly, in contrast to the new assay where there is a large distinction between the homozygotes and the heterozygous genotypes. So you can see with the original assay, we were getting 50% of the results right. This should really be percent match, not call rate. Whereas with the revised assay, the percent match was 100%. Okay, so here we show a table with the results of one of the runs that we did on the standard sample, and the key on the right is that the percent match with the reference profiles for F1 hybrid as well as inbred strains was 100%. The call rates were good in most cases as well, but some of them were a bit low. And, you know, that will, we're confident that will improve with increased usage of the mass array system. Okay, finally, here's, this is a table showing the results of parallel testing of field samples. They had been tested by our previous system, and they were retested by the Agena mass array. And if you look at the call rates and percent match, I don't have the data from the other system, but they're completely comparable. So we're getting the results that we expect for these inbred strains when testing field samples, and that's very important. When we do larger panels, we'll actually do spec readings and adjust the concentration of the DNA in the samples, but when we do routine quality control testing of inbred and F1 hybrid strains, it would be too laborious to do that, so we don't adjust the DNA concentration. Nonetheless, the Agena Iplex mass array had no trouble amplifying and correctly identifying the genotypes of these samples. Okay, the final comments. So for the standard and field samples, the Iplex SNP PCR results were comparable to those of the current assay system and matched our standard reference allelic profiles, but most importantly, all result calls were automated. So all the time we're spending reading results manually will go away. We can rely on the typer software to call the genotypes for the results. So the conclusion from this is that the Agena Iplex PCR mass array is a suitable replacement for the system that we're using now. And as was mentioned by Connie in the previous talk, we've really received very patient and superior support from Agena in terms of assay design, development, and troubleshooting, reagent preparation, which has saved us a lot of work, and then there is terrific literature that's available online. And I should have also mentioned here the great training that was available, and we've had that training more than once. I would say the mistake that we made in doing our qualification of this was not applying adequate resources to it. We have a very busy lab, so the time that was available to the technicians responsible for developing this platform in our laboratory was limited. And also at the beginning, I don't think we planned our experiments well enough. But aside from that, the transition to this new platform, to the Agena platform, has gone very smoothly. Agena has already developed for us, and we will work on qualifying next, a RAT32 SNP and 240 SNP panels, and mouse 384 and 128 SNP panels. The 128 SNP panels is specifically aimed at distinguishing among B6 substrains. So I want to thank all of you for your attention, and I turn it back to Christy.
Thank you, Dr. Scheck. And next up is Ms. Hunt. Please go ahead.
Thanks, Christy. So we just heard from Connie and Bill about some of the custom panels they've been able to implement into their routine testing. So I'd like to take a few minutes now to tell you about the options Agena offers in terms of custom assay development. Agena offers two options when it comes to custom assay development. The first is our online assay design suite. This tool allows you to input RS numbers or actual sequence and generate a design yourself. The second option is for you to use our Assays by Agena service, which I'll discuss further in just a few minutes. Here's a screenshot from our assay design suite. This online designer allows you to design standard genotyping assays, somatic mutation assays, or our ultra-sensitive liquid biopsy assays, which we call UltraSeq. Once you select the type of assay you want to design, you'll be taken to the main designer page. Here, you'll be able to input your RS numbers or sequences. There are several settings that can be modified depending on the multiplex level you're looking to achieve. In this example, we have the multiplex level set to 36. After inputting your RS numbers, you can begin the run. The software will then cycle through the 5 steps listed on the right. During the first step, it will blast the RS numbers using dbSNP and retrieve the sequences for each RS numbers. If you have used an input file containing a sequence, this step will be skipped. Next, the designer will look for any proximal SNPs and annotate them in your sequence so that it can avoid designing primers over a SNP. During the third step, the designer will identify the optimal placement for PCR primers by checking for variables like GC content and primer TM. The fourth step is where the designer actually begins multiplexing the different SNPs. It will check to see which SNPs can be placed together in a well and which ones need to be separated. The last step involves blasting the primers against the whole genome to ensure that the primers are specific and are amplifying only the intended target. Once completed, the output file that's generated will be ready to be imported into our typer software. You'll also receive an output that lists all the primers you'll need to order from an oligo supplier of your choosing. If you'd prefer to avoid doing any design yourself and would rather have one of our scientists build your panel for you, you can use one of our Assays by Agena services. The Agena team has extensive experience with developing a wide variety of panels, from oncology to pharmacogenetics to hereditary genetics. If you decide to go this route, you would just need to provide your targets to us, and we would perform the design, order the primers, verify that they're working as they should, and will then deliver ready-to-use primers to you. As I mentioned previously, the mass array system allows you to run many different applications from PGX to somatic mutation. We also have different sensitivity levels depending on the chemistry you'd like to run. Our standard genotyping application is called Iplex Pro and allows for detection of germline SNPs. This is great for PGX or hereditary disease testing and can also be used for somatic mutation testing in solid tumors. The limit of detection for Iplex Pro is generally around 5 to 10%. If you need greater sensitivity in your somatic mutation panels, we offer Iplex High Sensitivity. This allows for a limit of detection down to about 1%. And for groups that are performing liquid biopsy testing, our UltraSeq application allows for detection down to 0.1%. And if you're doing something completely different, we also offer mass cleave application, which allows for DNA methylation analysis. Here we see a typical workflow for running a test, starting with the assay design. This is a PCR-based approach, so as with any other PCR-based test, you would first need to collect and isolate your DNA samples. From there, you would perform the multiplex PCR, followed by the shrimp alkaline phosphatase cleanup, and then a single base extension. Following this, your PCR product is transferred to one of our spectra chip arrays and fired on our mass spectrometer. You would then perform your data analysis using our Typer software and reports, and the entire workflow can be completed in a single day. Agena has a large number of off-the-shelf panels available. Here's a list of some of the PGX panels that are currently available. Our most popular PGX panel is called PGX 74 and consists of 3 multiplexes that cover 20 of our most commonly targeted PGX genes. We also have more focused panels if you're looking at a more specific target like CYP2D6 or 2C19. Here we can see the genes that are part of our PGX 74 panel. One thing to note is that we can target SNPs, indels, and copy number variation all in the same reaction and workflow. Our C and B assays even allow for hybrid allele detection. We also have an extensive portfolio of hereditary genetic testing assays, such as cystic fibrosis, Ashkenazi Jewish inherited disease, celiac disease, and beta thalassemia. If you're looking to perform oncology testing, we have a broad range of pre-designed panels here as well. We currently have 4 liquid biopsy panels for lung, colon, melanoma, and targeted EGFR testing, and 3 high-sensitivity panels for lung, colon, and melanoma. We also offer a variety of solid tumor panels. The nice thing with all of these is that we can easily modify any of them to create a custom panel if there's a marker that you would like to remove or additional ones that you want to be able to analyze. Here's some other panels that are currently available. Our sample integrity and composition panels can be used as DNA QC, as well as a sample fingerprinting tool. And we also offer a blood group genotyping panel and several pre-designed methylation panels. I hope I've been able to show you that the mass array is a powerful, flexible platform that can be used to interrogate a wide variety of samples on an ever-growing number of genetic tests. And with that, I'll hand it back over to Christy. Thank you.
Thank you, Ms. Hunt. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. And we'll now start the Q&A. The first question is actually for all 3 of you. Priscilla, what is the extent of automation associated with your mass array systems? And then after that, we have a follow-up for Connie and Bill. How have you been able to manage scheduling between high volume and low volume assays on the mass array system, as well as integrating new assays into existing scheduling? So Priscilla, would you like to start?
Sure. The upfront workflow is just standard PCR, so you can use normal automation for that. And then once you've completed the PCR steps, our system is fully automated to take your PCR product from the plate, dispense it onto the spectra chip, put it into the mass spec, and then start the run.
Great, thank you. Bill, would you like to take the scheduling question?
Well, we're a high-throughput testing laboratory, and the fact that this assay can be completed in a day and does not take longer than the assay we have been doing means that scheduling this testing is no different from scheduling the testing that we're now doing. But we have our own laboratory information management system, which handles a lot of the scheduling of the testing that we do too. But in general, there's nothing about this assay that complicates scheduling compared to the other testing that we do.
Great. And Connie, anything in your experience?
I think my answer would be a combination of both of them. We are not high throughput necessarily like William for our SNP-based assays. However, we juggle multiple different assays within the same time frame. And so what we do is we set up the PCRs up front using our LIS, our LIMS system, to identify which samples are going on to the different plates, and we set up our PCR up front based on the samples that we're testing for any given assay. and we carry on through the assay as if it's one single 96-well plate. And then, like Priscilla said, the plate gets loaded onto the mass array. Everything is done automatically, and we use the Typer software to be able to identify which samples are going to be analyzed with which assay. And so we're able to make it, to combine everything in a single run on a daily basis.
Great, thank you. Connie, can you clarify for us really quickly how many targets you fit into each well, how many SNP targets?
Yeah, anywhere between 25 to 35 per well is typical for us. I think that it does depend on the size of the products you're amplifying. Priscilla would probably be better to answer that, but it does depend on the size. Because our targets are very specific, then we quite easily get anywhere in the averaging about 30 per well.
Okay, great. And Priscilla, we actually do have a follow-up for you. Our questioner wanted to know how does Agena actually fit so many targets into 2 wells? Because they specifically said their assays only have 6 to 8 at most. So they wanted to know how Agena fits that many.
We can fit, like Connie said, between 25 and 30 SNPs into a single reaction without any problem, because our mass window is broad enough to be able to fit all of those different products into that. If you're talking about a somatic mutation panel, then we have to limit it to about 10 to 15, just because we need to have more separation between each of the products. But for standard genotyping, it's no problem to get 25 to 30 in there.
Okay, great. I'm afraid that we've run out of time for today. We'd like to thank Connie Lisle, Bill Shek, Priscilla Hunt, and our sponsor, Agena Bioscience. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.